quality assurance for the marine science study program and aquatic resources management study program. Besides 8 laboratories in the Tamalan Ria campus, we also have a marine station which located in the island of Baralompo, a fish pond in Makassar City, a fish pond also in Baru Regency, a new campus in the island of Selayar, where we conduct a vocational study program, collaborating with the local government since 2021. The vocational study program itself is considered an example how to effectively reach out, especially our young generation of low-income fisheries community that live in faraway islands, and to offer them proper quality of education in the field in accordance to sustainable development goals. And Hasanuddin University is now recognized as a world-class university according to the QS World University ranking. Ladies and gentlemen, finally, in the end of this welcome speech, I would like to thank Professor Adi Maulana for the opening speech, and the event coordinator, Dr. Jamaluddin, and his team for great efforts on making this event happen. Special thanks and great appreciation to Professor Lawrence for the presentation and for spending the time and energy to share the knowledge and experiences with us. Hopefully, we could continue our collaboration in the near future. And for all participants, enjoy the webinar, and I wish you all productive and successful day. Thank you very much for your cooperation.
Thank you very much for your remarks. The following remark is from the Vice Rector of Partnerships, Innovation, Entrepreneurship, and Business, Universitas Hasanuddin. To Professor Dr. Eng Adi Maulana STMPhil, the time is yours.
Okay, thank you. Very good morning, everyone. Dean of Marine Science and Fisheries Faculty, our respected speakers today, Professor Lawrence Manzano Liao from Hiroshima University, and all participants in this Top Scientist webinar series today. Thank you very much for your time. Today's topic is Seaweed as a potential indicator of local environmental change. So this topic is a very relevant with UNHAS in two aspects. The first is easy, as what Mr. Dean has already mentioned before, that Sulawesi, where UNHAS is located, is one of the places who produce seaweeds in Indonesia. And in fact, Indonesia is the second producers of seaweeds in the world after China. The second aspect is UNHAS vision and mission. As we know that UNHAS is one of the biggest university in Indonesia, which concentrate in maritime continent, including the marine resources. And we are talking about marine resources. Of course, one of them is seaweeds. And today's topics is very interesting, because we are going to talk about seaweed as a potential indicator of local environmental change, which is, I think this is one of the biggest issues that we face today in the world. Well, thank you for Professor Lawrence for your valuable time to share your knowledge with us. And I do hope that we will have a fruitful discussions. And also thank you for the organizing committee. I know that Faculty of Marine Science and Fishery is one of the most productive faculty in Hasanuddin University to hold a webinar and other academic event, both international and national level. Well, on behalf of Hasanuddin University, formally, I want to open this webinar, and I wish you all the very best time and fruitful webinar. Thank you, and good morning.
Thank you very much, Professor, for your delightful remarks. Our distinguished ladies and gentlemen, the next agenda is the main agenda, which is divided into two sessions, the lecture and discussion. This webinar will be delivered by Professor Lawrence Manzano Liao from Graduate School of Integrated Sciences for Life, Hiroshima University, and Jamaluddin Fitra Alam, PhD, as the moderator today will guide the webinar and discussion. To our distinguished speaker and moderator, the time is yours.
Okay, thank you, Ms. Aliani, as the Master of Ceremony. The Honorable Vice Rector of Partnership, Innovation, Entrepreneurship, and Business Universitas Hasanuddin, the Honorable Dean of Faculty of Marine Science and Fisheries Universitas Hasanuddin, the Honorable, the invited speaker, Professor Lawrence Manzano Liao from Hiroshima University, distinguished guests, students, ladies and gentlemen, good morning. My name is Jamaluddin Fitra Alam. I'm a lecturer at the Faculty of Marine Science and Fisheries Universitas Hasanuddin, and today I'm acting as your moderator for this Top Scientist webinar hosted by our faculty and also hosted by the World Class University Program of Universitas Hasanuddin. Ladies and gentlemen, today we are delighted because we have an amazing invited speaker, Professor Lawrence, who I see already joined us in the Zoom meeting. So let us greet Professor Lawrence. Good morning, Professor Lawrence. How are you doing?
Good morning. Good morning. Thank you very much. I'm okay. Thank you. Look forward to this event.
Okay. Thank you, Professor Lawrence, for joining us. So we are excited to hear your presentation later. But before that, I would like to read your short biography as an introduction to our participants today. So, ladies and gentlemen, Professor Lawrence holds a special appointment as professor in the Graduate School of Integrated Sciences for Life at Hiroshima University since 2008. Professor Lawrence teaches general biosphere science courses and conducts practical workshops on the use of English in science communications. Professor Lawrence was awarded a Doctor of Philosophy in Biology with major in seaweed biology by the University of North Carolina at Chapel Hill. He spent a year after that at the National Museum of Natural History in Washington, D.C. under a Smithsonian Institution Educational Outreach Fellowship grant to study the seaweed flora of the Cuyo Islands, Palawan. His research interests cover a broad field of seaweed biology, from taxonomy and systematics to cultivation ecology and environmental bioremediation, which resulted in over 50 research papers published in peer-reviewed journals. He is now currently the editor-in-chief of the Philippine Journal of Systematic Biology and was formerly president of the Philippine Association of Marine Science and immediate past president of the Association of Systematic Biologists of the Philippines. In 2005, he received the Most Outstanding Alumnus Award in the Marine Science given by the University of San Carlos, Philippines. Also, Hiroshima University recognized him with the Excellent Educator Award in 2016. Ladies and gentlemen, that's the short resume of our amazing invited speaker today. He still has a long achievement, but if I read it all, probably the time is not enough. So, ladies and gentlemen, I believe that we will have a fascinating discussion later after the talk of the Professor Lawrence with the title Seaweed as Potential Bioindicator for Local Environmental Change. Ladies and gentlemen, without further ado, please welcome Professor Lawrence. Professor Lawrence, time and screen is yours.
Okay. Thank you. Thank you. Thank you very much. Thank you, Dr. Maludin, for the kind introduction. And thank you for inviting me. And I'd like to thank the Vice Rector for Partnership and Innovation, the Dean of the Faculty of Marine Science and Fisheries for facilitating this event this morning. I am very happy to be part of the webinar series today and share my knowledge about a research topic that I am very familiar with, I'm very interested in. Okay, it may be interesting for you to know that Hiroshima University, specifically Graduate School of Integrated Science for Life, and Hasanuddin University, we have an MOA, and this one will hopefully become a productive relationship between our two institutions. So I hope my talk today will serve to encourage you to pursue further the different agenda that we are going to do in the future. There are many areas of research that we can definitely do, and one of them is the seaweed biology. As already explained by Dr. Jamaluddin, my research interest is on seaweed biodiversity, assessment, ecology. I also interested in cultivation ecology and seaweeds, and very excited to be with all of you this morning because already we know Indonesia is one of the top, if not the top, producer of tropical seaweeds, at least the tropical seaweeds. It's number one in the whole world. And I think we should further support research about seaweeds and to make it a top commodity and a very good foreign exchange earner for your country. My talk, however, will focus more into the basic aspects of seaweed biology. And I will talk about this seaweed as a potential indicator of local environmental change. Okay, so I suppose you can see my screen now. And this is my address, and this is my email address as well. If anyone would like to contact me, I'd be very happy to hear from you. I'd be very happy to help you in any way. And if you are really interested, we can pursue some collaboration. Actually, it's very, very welcome. Okay, so let me start by showing this screen. That this is a map. This is a map of Indonesia, and as I've said, it's the world's largest seaweed producer, specifically tropical species. Indonesia is the largest archipelago in the whole world with 13 or 14,000 islands, largest group of islands in the whole world. And here in this map, you can see the red dots. The red dots indicate the areas of seaweed production. And I'm happy to show you here, Makassar in southern Sulawesi is indeed the top in Indonesia, and probably the whole world is the largest producer. And the government has already identified other regions in other provinces in your country as potential seaweed producer. Okay, south of Makassar, Nusa Tenggara, there's a lot of these things, and then Java a little bit, and then to the north, Northeast, Maluku, and Sulawesi Tengah, and Kalimantan Utara. So these are the existing ones. However, in the near future, the government of Indonesia wants to expand this one further. And like in this case, the target would be indicated by the yellow bubbles. So the red bubble of Makassar will further be enlarged into a really very big bubble. And the others also like Sulawesi Tengah, Sulawesi Tenggara, and other places. Maluku, for example, is also targeted. Maluku Utara and Papua Barat. Okay, so here this is very, very promising. And then to the western side of the country, Sumatra, Aceh, all of that, these are all targeted. So with all of these programs in the future, if this will all be implemented, no doubt about it, Indonesia will be truly the #1 in the whole world. So this is just to show you that these are the two major crops. I think you know this one, Capophycus, Eucuma. It is one of the most important, if not the most important, tropical seaweeds coming from Indonesia, with hundreds and hundreds of applications in the food industry, in the different industries, and generating a lot of dollars. Now, so far in Indonesia, the focus has been on these two species with food and industrial applications. Okay. However, I'd like to point your attention to other areas of application, such as agricultural applications. Many of these seaweeds, aside from Eucuma and Capophycus, mainly brown seaweeds, people have used it as fertilizer to help boost the farm productivity in tropical areas. So they have this seaweed extract powder. Animal feeds are also very important for agricultural application. A lot of the seaweed species have been incorporated into animal feeds to provide additional mineral sources. Now, this animal feed industry using seaweeds is becoming a very exciting field nowadays, because research has shown that some species of seaweeds, when they are mixed with the food for the cow, it can cut the release of methane to the environment. And we know that methane is a kind of greenhouse gas that is blamed, is the cause of global warming. So this is a very exciting field of research using seaweeds to feed the animals so that the animals will cut their emission, their release of methane into the environment. Another field that is also gaining interest is medical application. Seaweeds in many, many countries have been used for their nutrients, like in the case on the left side, fucoidan. Fucoidan is a multi-million industry in the pharmaceutical industry, and this is used for certain antioxidant, okay? And this is, it prevents cancer, supposed to be, it can help your immune system, and so forth and so on. On the right side of the screen, you can see that some seaweeds have been used in many new products. For example, there are studies now that use the seaweeds for making contact lenses. Okay, in that picture there, contact lenses, intraocular lens. Intraocular lens means these are small lens that you can insert into the eyes of the cataract patient. You can also do that. And then another polymeric product is the biodegradable suture. This is also gaining a lot of attention suture is when you have an accident, you go to the doctor and you have a wound, they want to sew your wound, okay? They don't use the thread anymore. They use the thread coming from the seaweed, which is biodegradable. After a few days, it will dissolve. It will dissolve at the same time that your wounds will get healed. Okay? Wound dressings also is very important source, especially in North America. A lot of the seaweeds are used for making all these sterile patches that you apply to the wound. And of course, in that picture here, you can see capsules and tablets. They are all made of seaweed extracts. So a lot of medicinal and medical applications. Seaweeds also have environmental applications. Many seaweeds are used as monitoring tools, and I will talk more about that. Certain seaweeds, when they are abundant, they can tell you something about the environment. Usually it's a warning sign, it's a sign of danger. So this is for monitoring. Aside from monitoring, some seaweeds can be used also for remediation. Remediation means try to clean up the environment. And one of the processes of remediation is called the biosorption. So some seaweeds, when they are added to water, they can absorb heavy metals. So if your marine area is full of heavy metals coming from factories, coming from household, domestic waste, some seaweeds have the capability to do a biosorption of heavy metals. Okay, so the monitoring of environment can be done either using living samples, as shown on the right side of the screen, or they can also be using preserved samples. So for preserved samples, okay, you might be asking, oh, they are already dead. How can we monitor the environment? Well, the answer there is the dead ones, the dead samples can give you an idea of the environmental condition at the time that they are still alive. So if your sample was collected 100 years ago, okay, and you preserve that sample and you can still see that sample 100 years after, then you can look at the sample and you can get an idea what was the situation of the environment 100 years ago from that place where the sample was collected. So that's why it's very important that we preserve these samples very well, and we can use these samples for checking the environmental condition from the past. Now, this is a process that is not only in seaweeds, but also in many other plants, okay? Now, these plants are preserved inside a museum. Specifically, we call it a herbarium. Herbarium. So what is a herbarium? Herbarium is like a specialized museum. It is a repository of plants in the broad sense, including algae, seaweeds, including fungi. So a picture of herbarium is there on the screen, and usually very easy. You just press all the samples flat, and then you label, and then you keep everything inside the cabinet, and it can preserve for 20, 50, 100 years of herbarium. Now, the herbarium samples are mainly used for taxonomic research. So we check the morphology, we check the inside, the anatomy, and we can get some idea about the ecology of the previous time. This is very easy. That's why I'm talking about this to you, because this is something that is cheap, requires low technology, and is very relevant for a country like Indonesia. Okay, it employs very easy techniques, but sometimes laborious. Laborious means need a lot of help, need a lot of people doing it. Okay. Also, the people who are trained to do this technical work with skills for herbarium work, sad to say, they are becoming little by little, they are retiring. Why? Because and very few young people can come in. Why? Because maybe it's not well known, maybe it's not so interesting. Yeah, maybe there's no money there. Okay, but I can tell you that this herbarium is very important for research. This herbarium is very relevant, especially now that we are subjected to climate change, to environmental change. So these herbarium samples can be used for monitoring previous situation and compare it with the present situation. Yes, I said this already, that if the samples deposited in the herbarium are very well preserved, you can get a lot of useful data. So I want that at the end of this talk, you will be encouraged, and maybe your university can put up something like this as one of the research resources that's available for faculty members and for students. Right now, the herbarium is also undergoing some changes. So because of social media, because of good technology, so there is now what we call the virtual herbarium. There is now what we call the digitization. So everything is digitized. Everything now is turning into the digital media. Okay, so we will show you that one. Now here are some pictures of herbarium, and as I've told you, these herbarium are very simple. You know, just put some cabinets and then paper, and these things are very easy. We can do it easily in your country. Okay, we cannot say that, oh, we will not do this because it's very expensive. Oh, the equipment is very advanced. No. This is very easy, very simple, and very useful. Now here is one sample of herbarium. Okay, and the herbarium is like a sample. You press it flat and you stick it into a paper like this and complete with different labels. Okay, and all these labels are very important information. Where are they from? When are they collected? Okay, when are they collected? What is the color of this one? You know, something like that. So the herbarium is a record. It's a record of an occurrence of a particular plant at a particular place, particular time. Okay, so it is a verifiable record. Verifiable because you can always go to the museum and check the sample, and the sample is there as your evidence. Okay, it's unambiguous. Unambiguous means it is very clear. Okay, very clear. It is really the real evidence. So the sample is permanent. If you preserve it, some samples can last 100 years, 150 years, 200 years. It can last very long time. And then the data inside the herbarium can also last for a long time. So it is a very practical tool for recording some of these species. Recently, the herbarium is used for climate change studies. Okay, climate change studies. As I've said, some of these herbarium date back from many years. So at the time when these samples were still alive, they are subjected to the environmental conditions found during that time. So these material are a good source of proxy data, okay? Proxy data to find out about the past conditions in your area, where these samples come from. So sometimes it can also be used, especially among the flowering plants, okay? you can use it for tracking down phenological changes. Phenological means at what time does it have a flower, at what time does it have a fruit, at what time is it like this and like that. So it's very nice to use as an evidence how are these different fruits and flowers affected by climate change. So some of these structures inside of plants, like flowering plants. Let me talk about flowering plants first, okay? Many of these structures in the flowering plants can be changed, can be affected by changing environmental conditions. Let me show you an example. We are all familiar with stomata, okay? Stomata are small holes found on the leaves, okay? So these stomata are small holes where the oxygen enter, no, the CO2 enter and the oxygen comes out. Okay, I think we are familiar with this one when we take biology in high school or even in university, stomata. So one can go to the herbarium and check the stomata of samples collected from 20 years ago, from 30 years ago, and so forth and so on. So here, using the microscope, you can see the number of stomata. So on the left side of the diagram, you can see that if the stomata are very few, okay, very few, okay, can you see the stomata? They are encircled in the red line, okay? So when they are few, it means normal CO2, normal CO2 level. But then if there are many stomata there, it means the CO2 is low. Now, if you try to relate the sample that you found in the museum sample, the low stomata density, if there are very few stomata density, it indicates a high atmospheric carbon dioxide. So high in atmospheric carbon dioxide in this diagram indicates warming global temperature. Okay. Now on the right side of the screen, if the stomata density is high here, it indicates a low atmospheric CO2, which indicates normal or low global temperature. So this is very nice. You can connect the global temperature of 30 years ago, 50 years ago, by examining the stomata from the samples collected 30 years ago, 50 years ago. Very nice study. Another study, this is from the United States. They look at the museum samples, 372 samples representing 37 genera collected from 1885, very long time ago, and between 1885 and 2002. What did they find? They found that the flowering time of these different samples became earlier. So what does that mean? Normally, if the flowering time is April, but then they found samples that are flowering in March. So that means, oh, normally it's April, now it's March. So from 1980 to 2002, they found that the samples that they collected produce flowers 8 days earlier. So something wrong with the climate, something wrong with the temperature. So they try to connect what they found in the flower with what they found in the climate temperature data. So they found out that from 1980 to 2002, the city has experienced 1.5 degrees Celsius increase in the annual temperature. So they said that every year from 1980, the temperature slowly go up. So as the temperature goes up, it means that the flowering of these plants become earlier. Okay, so this is one example. Another example is they try to connect the flowering time of sample, in this case, 87 species from Arizona, collected from 1902 to 2006. And they found that 19% of these species, 19%, show advanced flowering. Some of them show delay flowering. So something is wrong. It's not normal anymore. So if the normal is April, some of them flower in March and some of them flower in May. So something is anomalous. And also they show that between the rural area in the villages compared with the city environment, they found that there is a difference of about 28% in the flowering time difference between urban and rural areas. So change was very clear in this by looking at the samples found in the herbarium. So another nice study, and I think this is very interesting study, they collected the same species of sample from 1876 and compare it with 1913 sample and compare it with 2002 sample. So what did they do? They collected pollen grains. Pollen grains, these are very small things on the right side. These are the food for butterflies, the food for the bees, the food for different insects, and they are full of nutrient. Now they found out that the protein content of the pollens went down, decreased when the CO2 in the air increases. Very, very interesting. Okay, so from 1876 to 1913 to 2002, then when they analyzed the pollens, the protein went down. The protein went down. So it is connected to the increase of CO2 in the air in another research. Now, why is this so important? Because the pollens are the food of these bees, right? And the bees are the pollinators. So if the pollens will go down in terms of protein content, the population of bees will also go down, and it is not very good for the farmers. Okay, so in this particular study, they connect the herbarium samples with protein, the protein content of the pollen connect with the bees, and the bees can connect to farm productivity. Okay, very good. It's the same thing as like this. No? I'm just showing you this. When you cut a tree and you can count the rings. Now, the rings of the tree can tell us many stories. In this particular example on the right side, you can see that this tree was planted in 1913, 100 years ago. But then from 1913 to different years, 1918, 23, 33, 36, 39, there are histories, okay, which are documented by these tree rings. So I'm showing this to you because I want to tell you that the samples kept inside the museum can tell us a very important story. So the oldest herbarium in the world, started almost 500 years ago in Europe, okay? 1532. Today, there are 3,000 herbaria or museums around the world, okay? 3,000. And in these 3,000 herbarium, the total sample is about 350 million samples. The oldest one is 400 years old. Research has shown that even for samples 200 years old, one can get DNA from these samples. Imagine you can get DNA from a 200-year-old preserved plant. Okay, this sample on the right side is collected 1882. Okay, almost 150 years old. Still very nice. still looks interesting, yeah. But then if they get some DNA from here, they can get that information. Now, this one is the database on all the herbarium around the world, okay? And this is maintained by the New York Botanical Garden. In case you're interested, the link is there written down at the bottom of the screen. And this is a worldwide index database of 3,100 herbaria around the world. The largest collection in the world is in Paris. How many? 9.5 million samples in the National Museum in Paris. In Japan, about 5% of universities have museums. The largest one, University of Tokyo, 1.7 million samples. In Southeast Asia, not so many herbarium. The largest one is in Indonesia. Okay, we should be proud of it. In Bogor, it's called the Herbarium Bogoriense, and it is the largest in ASEAN, and it has 2 million samples. 2 million samples. And the last sentence here, I just said that many of the countries in Southeast Asia are suffering from biological colonialism. What do you mean by that? For example, okay, somebody wants to study the fish of Indonesia. Okay, so if you want to study the fish of Indonesia, naturally you want to go to Indonesia and try to collect sample. But then somebody will tell you, no, don't go to Indonesia. All the samples of fish in Indonesia is not in Indonesia. They are all in Netherlands. Why? Because the Netherlands was the colonizer of Indonesia for a long time. So this is what I mean by biological colonialism. So for example, in my own country, I'm from the Philippines. If somebody wants to study the flowers or the plants of the Philippines, we don't go to the Philippines. We go to New York Botanical Garden, or we go to Europe. Why? Because the largest collection of flowers and plants from the Philippines is in America, because America was the colonizer of the Philippines for 50 years. So this is what I mean. So one of the messages in this particular presentation is that I want to encourage Southeast Asian countries to empower themselves, okay, to take charge of biodiversity studies. Because right now, it's the foreign countries, it's the Europeans, Americans who are taking charge of the biodiversity in ASEAN. Okay, this is sad, but this is the reality. Okay, and this is not impossible. We can do it, starting with the young people, starting with the support of the different institutions. I look at the database in the New York Botanical Garden, and I found that these are the list of the herbarium in Indonesia. So there is from Java, Bogor, Yogyakarta, Palu in Central Sulawesi, Bandung, okay, and Manokwari, Padang, Malang, Chibinong, okay, Kalimantan, no Makassar. So this is your challenge. I want you in the future to be the leader in Makassar and try to build something that you can be proud of. Okay, so let's go to the seaweeds now. So these are pictures of seaweeds, and these are herbarium samples. And very easy, just collect the sample, a nice sample, and then just press them and then put them into the white paper. And you can use this one for your research. You can use this one for your herbarium. Okay, so the samples are underappreciated. Okay? Many people don't pay attention to that. I can understand. Everybody wants to go to the seaweed culture. Because that has a lot of money. okay, especially in Makassar. Makassar is the largest region, producing region in your country. So I understand that all the focus of research is on this, okay? But I also want to call your attention to focus on biodiversity, especially in environmental monitoring. I mentioned already that the herbarium is easy to build. It does not cost a lot of money. You only need the easy and low technology. You only need a low maintenance cost. I think this is really very interesting for you. Now, we know that seaweeds are mostly under the water. Okay? They are submerged. And because they are submerged, the seaweeds are influenced by the water. So if something happens to the water, if there's a pollution in the water or some kind of environmental change in the water, naturally, it will be affecting the different seaweeds. Now, during this time, if the temperature suddenly changes in the marine environment, so the seaweeds are directly affected because the seaweeds cannot run away. The seaweeds cannot swim away, unlike the fish and other animals. So the seaweeds had to stay in one place and they are subjected to pollution, high temperature, low temperature, high salinity, different environmental changes. And we know that if the temperature increases, just like this one, it can affect so many other physiological processes. Okay, if you have a high temperature, you can have stress, you can have a water loss, you can have changes in photosynthesis, so many, it can affect the growth. So if there are impacts like this on the seaweeds, and these seaweeds are stored in the museum, then you can see that these museums are useful in telling the story about the past, about the historical changes. So we know that if the vegetation shifts, okay, if the temperature, if the seaweed suddenly changes, it can affect the different animals. It can affect the different aquatic organisms because we know that the seaweeds occupy the base of the food pyramid, the food chain. Okay, so here it is. Okay, sorry. Let me see. Let me go back. Oh, sorry. Yeah. Okay. So if you see the arrow from the when there is environmental change, you have a vegetation shift, and then the arrow to the left connects with aquatic organisms and all the microbial and organic matter cycling, etc. It will all affect all of these. It's a chain reaction. In one example that I show you here, this is a very nice research about herbarium samples of seaweeds in Australia. So in this particular research, the researchers examined 20,000 different herbarium samples that are collected around Australia from 1940 to 2009. And what they found out is that many seaweeds from the north are moving to the south. Okay, this is Australia. So many of the seaweeds are moving to the south because the south is becoming warmer. So this is an evidence that the seas around Australia are becoming warmer and there is some kind of environmental change. So the environmental change have been facilitated by the currents that push the thermal barriers. What's a thermal barrier? Thermal barrier are these red dotted lines, okay? So the red dotted lines on the left side, you can see 22. So the temperature 22 is there. And then the temperature 20 is down here. So these 22 is push further going south. So the 20, the line of the 20 becomes now 22. So therefore, many seaweeds from the north go down to the south. The same way on the right side of the map. The 22 line is pushed down to the 20 line, is pushed down by this purple arrow, which represents the warm current. Okay, now this one is just a review, a quick recall. Some of you know this one already, the different life cycle of algae. Now, the first type of life cycle is what I call isomorphic. Isomorphic, meaning to say the haploid and the diploid, they look very similar, isomorphic. So we call it the I type. The second one here is called heteromorphic. Heteromorphic means the gametophyte or the haploid and the diploid are very different. They are very different, but they belong to the same species. Okay, so there's an H type, there is an I type. There is also another seaweed that has no cycle. Okay, now why are we mentioning H type and I type? Because there was a study in Japan, 2012, 10 years ago, wherein the researcher used to classify the I type and the H type and get a ratio of the I type over the H type. And they look at the different samples in the herbarium samples from 1914, okay, 100 years ago, more than 100 years ago, until 2009 in one area in Japan. And then they used the IH and try to calculate the ratio. They call it the floristic index. And they found out that the floristic changes because of the I over H correlate with the rising trend of yearly mean surface temperature in winter. Meaning to say the winter is getting warmer in this place in Japan. Why are they, okay, why did they say that? Because the ratio of IH is a measurement of how much global warming or temperature warming is happening. So this is a very nice example that you can also do in your country. Another one is this one. This is a very alarming situation now in not only in Japan, but also in many parts of the world. And this is what we call tropicalization. Tropicalization simply means warming of the sea. The cold water sea gradually becomes warm, and this is called tropicalization. So the tropicalization in one area can be documented by examining different herbarium samples. Now, in this particular study in 2014, they monitor the herbarium samples collected from 1990 to 2013. And they also went the scuba diving and they look at the environment. And here on the left side of the map, you can see that there is a current, a warm water current that goes from the south, from the Philippines, go to Taiwan, to Okinawa, and finally it goes to Japan. It brings the warm water. So in this site, on the right side of the screen, you can see that the seaweed has changed. So the one on the upper left side, this was observed in 1990. 7 years after, the seaweed start to disappear, and you have this picture on the upper right side. And then from year 2000, all the seaweeds are gone. And you can see that in picture lower left side. Okay, this is the same place. And then 5 years after this one, this is 13 years after the seaweeds have gone, the picture on the right side lower corner, you can see that it becomes not a seaweed forest anymore, but it becomes a coral reef already. So these are all documented by actual diving and at the same time documented by examining the herbarium samples. So in many parts of the world, these seaweed forests, or we call these marine forests, are disappearing because the global warming is coming and the seaweed forests in the red zones do not like the warm temperature, so they die, many of them. In Europe, in Japan, in North America, in Southern Australia, this is happening, okay? And this can be documented by looking at the different herbarium samples. Another problem, we know this one, ocean acidification, okay? I think you're familiar if you look at the diagram on the left side, you can see that when there is an increasing CO2 in the air, this CO2 will enter the water. Once it enters the water, if you follow this diagram here, it enters the water, it forms carbonic acid. Okay? CO2 plus H2O equals H2CO3. Now, this H2CO3 can be broken down into bicarbonate and hydrogen ions. And it can also become carbonate ions, okay? So this is the whole mechanism. So under high ocean acidification, under low pH, the pH value low becomes more acidic, the carbonate ion disappears. And what happens, many of the animals, like the shell on the right side, is unable to make some shell on its own. So this one affects crabs, shrimps, starfish, mollusks, oysters. Okay, they need the carbonate in order to make their skeleton, right? So with the slowly disappearing carbonates from the marine environment, then it becomes a problem. So among seaweeds, there are seaweeds that need calcium carbonate. These are called the coralline algae. So in a study in 2013, this researcher compared the sample of these species, the purple one on the picture. She compared the samples collected in 1981, 1997 with samples collected in 2012. What did she find out? Look at the picture on the right side. On the upper picture, you can see that this purple seaweed has very thick skeleton. This is the sample from 1981. Whereas the one on the right side, this is a sample from 2012. So this simply means that the available carbonate is already very low. There is already low supply of calcium carbonate in 2012 compared to 1981 when there's still a lot of calcium carbonate. And then you can see that the skeleton is thick. So by examining different samples coming from different parts of the world, coming from different years, and looking at the calcium carbonate, we can get a picture of the environmental conditions during 1981 or earlier, okay, and try to compare it with the present one. So this is one of the very nice application of seaweed samples for monitoring climate change. In this particular case, for monitoring carbonate content in the water. Okay. Seaweed samples can also be used for determining biogeographical boundaries. So this is a study in 2004, and what they did is they used the museum samples and they try to plot where they are. So using the museum samples, they found out that in this red line, they found many species that are tropical species. And then on the other side, there's this gray line, and they found out that the southern part of South Africa is really cold water. So where the red line and the gray line meet, this is considered as a biogeographical boundary, okay? And this is a very interesting study because it uses 100% herbarium samples and other museum materials. As I've said earlier, DNA can be extracted from samples even though they are 50 years, 100 years. Of course, the samples that are older, the chances for DNA is lower, okay? So if you are able to get DNA from a series of samples collected from different parts at different times of the century, you can use this one for plotting invasive species. Okay, there was a study in 2007. They found some invasive haplotypes, native haplotypes. Okay, and I think this is a very nice way of using the herbarium samples for studying molecular ecology. Okay, molecular DNA ecology. Okay, so this is one application. Another application, this one is from China. Okay, checking the species diversity of sample from 1933 to 1935, compare it with samples obtained from 1990 to 2009 from one island in China. And what they found is that the species number from 1935 and the species number of 1990 are the same. So there is no change in the number of species. However, there is a change in the species composition. Okay, can you see? Species number, no change. But species composition, the kind of seaweeds occurring in 1933 with the samples from 1990, from 2009, different, but they are the same number. So one of the things that they said is that there is a lot of coral reef degradation. The different status of coral reef the healthy coral reef, okay, of course can produce can host more species and can produce certain delicate species, and compare it with the coral reef that is already damaged, then what kinds of species are there? Maybe just the strong species, just the opportunistic species. So then the kind of species from 1933 to 2009, if you compare, it will give us a clue into the kind of coral reef environment before and the kind of coral reef environment today. Tourism infrastructure development. So they found out in this study that because of so many hotels, so many resorts, so many of these tourism infrastructure, they can also affect the kind of seaweeds that grow at a particular time. Aquaculture development. So when there's a lot of fish cages, fish ponds, naturally there's a lot of waste generated by aquaculture. This one can also affect the species composition from 1933 compared with 2009. So very, very useful. Very, very useful and very informative. Here is a study of one of my students in Philippines, and he was monitoring one of the bay. You can do this in Sulawesi. You can do this in Makassar easily, okay? So it happened that there was a collection from 1969 to 1970. And they found 11 species of red algae, 9 species of brown algae, 10 species of green algae. And then when these students compare it with 2013 and 2014, the number drop. The number drop. Okay, so this is one of the important findings. What kind of species were lost? Species that have large blades, large surface area, and these species usually are found in very clean, no pollution environment. Now, when you change the environment, like you put a lot of sediments, you put a lot of silt, you increase the sediment, it will cut the light that enters into the sea. So what happens? Photosynthesis will also decline. So these species cannot survive under this low light environment. So they are gone. They are wiped out. Another species that they found, the student found, are these two species. A lot of these species can survive even under eutrophic conditions, under conditions with a high pollution load. For example, ulva. Okay, I think you've seen this in your country many times, the green seaweed on the left side. Under high nutrient, under high pollution, no problem. This ulva can survive. How about the one on the right side, Adina species? Under high nutrient, under heavy metal pollution, no problem. It can survive because this sample is known to absorb heavy metals. It can remediate the water by absorbing the heavy metals. So in the study in 1969, they cannot find this species. They cannot find this brown species, but during 2013, in that same area, they found already this one, so many. So I think if you go around the area of Makassar, you can find this one, this same species there. What does that tell you? It tells you that perhaps there is a lot of pollution in this particular area. How will you prove that? Of course, you can make water analysis. You can do some monitoring using chemistry, using some other ecological factors. Another thing that was found in the study of my student is that a lot of these species, sargassum, for example, these sargassum, they have roots, okay? Now, if you have roots, you need some rocks so that they can attach. Now, what happened is that when he did this study in 2013, he cannot find any of these species, cannot find sargassum anymore. Instead, they found these kinds of samples that do not have roots, but they have some complex anchoring system that does not need any rocks. So it needs only muddy substrate. So it means that the disappearance of the sargassum and the appearance of the sample Halimeda on the right side means that the general habitat has changed from rocky into a muddy and clay and muddy bottom. So the change of substrate is very important because once you change the substrate, it can also change the species composition. Okay, if any one of you is interested, this paper is already published. I can send you a copy. So this is a sample from that same study, and there are 6 stations that were compared, and you can see that Adina, the one that can absorb biosorption heavy metals, is very common. It's present in all the 6 stations. Halimeda, the sample that I showed you with the old fast, like the onion, also very dominant. All 6 stations are also there present. In another survey in Brazil, okay, this is a very interesting study, which you can also do in your area. They compared the herbarium samples from 1957 to 1978 and to 1991 and 1998. So what did they find? In 1958, they found 100 species in this area in Brazil, 100 species. However, in 1978, the 100 species became 64. Ooh, that's not very good. But then when they survey again this area using the herbarium samples as well, they found that from 64, it became 91. Why? Because in 1998, 1997, they built a sewage treatment terminal. So meaning to say, the water quality in that area, it went down from 1957. It became very bad in 1978. But from 1978 to 1998, after building the sewage treatment facility, the seaweeds can actually recover. It did not recover to 100, but it recovered from 64, it became 91. And this is all supported by herbarium records. Now here is an example from Japan. I was lucky to know this husband and wife. This husband and wife, they are not scientists, but they are science teachers. They are teachers in high school. So what they did, they monitored the seaweed in one area in Hiroshima from 1954 to 2004. 50 years. They visit that site every month. Okay, it's their hobby. Husband and wife, they go there every month for 50 years. And what they did, they compiled all their information and they made it into a book like this one. Very easy. Just go to the field, collect, and then record the samples. And if you look on the left side of the screen, you can see that one example of the species, you can see the dark black bars. So these black bars indicate the presence of these samples. So in 1959, 1960, the black bars are there. And then after 1960, no black bars. Oh, mysterious, what happened? But they came back in 1987. It came back in 1988, and they came back until the present. So meaning to say, the history of this particular species is such that they are affected or influenced by the environment. Now look at this particular example. This is ulva. This is the green seaweed. So the green seaweed, they are abundant in the early part of the survey and from 1987 all the way until the present. Okay, it's there. It does not go away anymore. So this is what we call this one, the green type. And this is an indicator of pollution. Okay? So we call this a green type. So very simple, but this hard work. You have to go to the sea every month for 50 years. So you don't have to go there for 50 years. You can go in 20 years or any. Why? Because green algae like this one, okay, is sometimes considered as an index of aquatic organic pollution. Because I told you that this green algae, they are useful as index of aquatic organic pollution. The total number of organic pollution, you can use the simple formula, number of green algae divided by the total algal species. So if there are more green algae in your sampling site, then it is a danger sign that it could be because of the source of pollution. Now, you can go back to the herbarium records and you can dig through the records there and you can see, oh, are there many seaweed samples in 1950? How about 1960? How about 1970? How about 1980? You can make a story about the occurrence of seaweeds using the herbarium samples deposited in the herbarium, the museum. Now, this one, just to show you, I'm going to end my talk soon, that it's very important the seaweed vegetation can tell us a story. Okay. Now, it's very important to monitor the seaweed vegetation changes because we said that the seaweed supports the marine food chain. If something wrong happens to the seaweed, then it will affect the sea urchin, it will affect the fish on the top, it will affect humans because we rely on the, 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 the seaweed to support the food chain. So it will become a what I call the domino effect. So if something wrong happens here at the bottom, it will affect the next stage, all the way up to the highest stage. So today, there are so many, there are many studies now published in the literature about the use of herbarium samples, how these herbarium samples are used as environmental tools, as biogeographical tools. Okay? Now remember, there are 3,000 museums around the world in that database, remember? And how many in Indonesia officially recognized in that database? Less than 20. But all over the world, there are about 350 million plant samples. Each sample, each of those 350 million samples awaits examination. Many of those samples, all of those samples, have some message that's waiting to be discovered. Okay? So I think we should no longer have any excuses for not using these herbarium samples. I think we should start it. So the physical evidence, like the herbarium samples, cannot be intimidated, cannot be changed. The physical evidence, if it is well preserved, it is forever. Okay, just like when there is a crime happening, the police will gather all the evidence, and all this evidence will be kept so that in the future, the evidence will be available for further investigation. Okay, so this is, I like this quotation because it says the physical evidence cannot be intimidated. It cannot forget. The physical evidence is not forget because will not forget because the record is permanent. The physical evidence that sits there and waits to be detected. It waits to be preserved, evaluated, and explained. Okay? So I'm going to give you a very important message that I think it is about time. It is the right time to look at the herbarium samples beyond their taxonomic value because I have said that most of the herbarium samples are only used for biodiversity, they're only used for taxonomy. Yes, they are useful for taxonomy. This is the original one. But I think the herbarium samples should be examined further because we should appreciate them as containing valuable information from the past. So we should start appreciating, okay, this is my word, appreciating the value of herbarium specimens as storytellers of the past because they contain a lot of data, they contain a lot of information from the past. So they are very valuable as storytellers of the past and of the present. And it will give us a view into the future. Okay, so this is very, very important. I'd like to end the talk by giving you some information. Some of this information I think you already know. I mentioned that Indonesia is very lucky. It is the largest archipelago in the world, 13,000, 14,000 islands from the east to the west. You know, there's no other country as big as Indonesia. There's no other country as rich as Indonesia in terms of biodiversity. Now, you are lucky because many years ago, there was an expedition. This is an expedition organized by the Dutch, and it's called the Siboga Expedition. You may have heard of that. And in 1899 to 1900, the ship from Netherlands came to Indonesia, and they started a survey. So this is the place of the survey. They went around Makassar, Sulawesi, at the time it's still called Celebes, Sulawesi. They went to Maluku, they went to Papua, they went to all these places. Okay, they went to Timor, Flores, Sumba, Java. Okay, they collected so many samples. But because of this, the seaweeds and other marine organisms of Indonesia became world famous, because it's the first time that expedition came to Indonesia, and they called the attention of the world to the rich biodiversity, marine biodiversity of Indonesia. Where are the samples of the Siboga expedition? They are not in Indonesia. They are in Netherlands. Okay, so this is one of my points, that we are suffering from biological colonialism. Okay? Now, again, we're lucky. In 1984 to 1985, there was another expedition in the same area, Snellius II expedition. So they went to Sumba, Komodo, Flores, Sumbawa, Salayer, it was mentioned earlier, south of Sulawesi, and then they went to Ambon, okay, and different places. Where are the samples? The samples are in Netherlands. Okay. There was another sample. There was another expedition. This time, this was in Makassar, where Spermonde, this is 1993. Spermonde survey. If you look at the map, you can see Sulawesi, and Makassar, formerly known as Ujung Pandang, and the Sulawesi. This is a combination of Netherlands and Belgium, and some Indonesians. Okay, and in this particular survey, the researchers found more than 200 species. More than 200 species from 1988 to 1990. 30 years ago already. It's the biggest seaweed survey in the Makassar region. And it resulted to a publication in 1993. Now, why am I mentioning this? Okay, by the way, all the samples from here, Spermonde archipelago, they are in Netherlands also. But I read from the paper that some samples are left behind in the Bugor. Okay, but are there samples in Hasanuddin? Maybe not. I think you can do a survey and go to the Spermonde archipelago again and try to collect and compare your samples from 2022 with samples from 1993, and you can have a good story, and you can have a good publication from that kind of research. What are some of the challenges for seaweed biodiversity research in Indonesia? I already mentioned this many times. Taxonomic studies are very few, okay? The number of taxonomists, the number of Indonesian taxonomists, when you compare it to the actual size of the country, the ratio is very small, okay? In the whole of Indonesia, how many people are doing marine seaweed taxonomy? Less than 5. Active ones, less than 5. I know 2 of them, okay? One in the north of Sulawesi, in Manado, and then another one in the Lipi. That's it. No, very few. Number 2 challenge. I think the existing taxonomic data in Indonesia needs updating. Okay, if I ask you the question, how many species of seaweeds are there in Indonesia? I don't think anybody has the right answer. I don't think anybody has the correct answer because the information is scattered. Okay, very basic question. How many species of seaweed from east to west of Indonesia? I don't think anyone in Indonesia can answer. Maybe someone from Netherlands can answer, but within the Indonesia, nobody can answer. Number two, third challenge. Many of the data, I know in the last 10 years, there are small studies, very localized studies from Java, from one regency in one place, okay? Mainly in local journals and mainly in Bahasa Indonesia. So of course, we know that if you publish in Bahasa Indonesia, outside of Indonesia, nobody can understand. Well, you can use the Google Translate, fine. Huh? But you see, publications in English are more widely well known, but they are very limited. The paper that I show you from Spermonde is one of the few examples of Indonesian study published in English. The papers from the Dutch from 100 years ago, they are not in English. Many of them are in French, or maybe some of them are in Dutch. So it makes it more difficult to access the information. Specimen repositories in Indonesia, very few. Okay, I told you Indonesia is so big, 13,000 islands. How many herbaria, how many museums there are? Less than 20, less than 30, very few. And the few Indonesian museums may be understaffed or lacking technical expertise. Now, this is not an impossible job. We can train technical expertise, and it will take time, but it needs the effort, commitment of young Indonesian scientists, okay? If we can train young Indonesian scientists, and these young Indonesian scientists can do surveys, can do taxonomic study, oh, Indonesia 20 years, 30 years from now will become #1 in the seaweed biodiversity study. Same is true in the Philippines. The Philippines is also a big country. How many taxonomies in the Philippines? 6. How many islands? 7,000. One taxonomy for 1,000 islands. Too much. Yes, this one I mentioned already. Most Indonesian samples are deposited abroad. And because they are deposited abroad in Europe, they are largely inaccessible by Indonesian researchers. So I think to provide the solution for that, because it's impossible to bring those samples back to Indonesia. It's impossible to bring the samples from Netherlands back to Indonesia. Impossible. So the solution there, collect new ones. Collect new ones, because Indonesian natural resources belong to the people of Indonesia. So you collect the new ones and put all these samples in Indonesian universities and institutions. This is not impossible. This can be done with the support of academia, with the support of government. Okay, there is a need to establish more local museums, herbaria, supported by government, supported by academia. We can start in Universitas Hasanuddin. If you have not started it, we can start very easy. And in a few years, the museum will become a very good center for research for Faculty of Marine Science and Research, Fisheries, for example. Okay, so these are some of the challenges. There are many other challenges. Okay, so I hope with this presentation, I'm able to encourage you, administrators, young people, to do something about the use of seaweed biodiversity for research, use for environmental monitoring of environmental change. Thank you very much. Terima kasih. I hope I'm now willing to open to answer some of your questions. Thank you for your attention. Thank you, and I hope I was able to give you some new information, some encouragement, some enlightenment. Thank you very much, and I will turn over the floor to the moderator.
Really enjoyed your presentation, and of course I believe the audience also enjoyed your presentation, because that was awesome. Thank you very much for sharing with us about your experience in seaweed research, Professor Lawrence.
Yeah.
Now we still have time for discussion. So actually there were already some questions addressed
Yes.
to you, Professor
Yes.
Lawrence, in
Yeah.
the
Yeah.
chat column, but I would
Yes.
like to invite Dr. Widya Stuti, who already raised her hand in the middle of your presentation. So probably she was curious about some of your talks.
Yeah.
So
Yeah.
let me invite Dr. Widya, please.
Hi, Dr. Jamal. Thank you very much for the chance, for the time. Good afternoon, Professor.
Good afternoon.
Yeah, it's a really insightful and inspired thought to you, from you.
Thank
Yeah,
you.
yeah. In the middle of your presentation, I'm really curious about the previously you mentioned about the potential DNA work for seaweed herbarium that have 200 years
That's
age,
all. Yeah.
if I'm not mistaken.
Mm-mm-mm.
Yeah. Uh, do you have like a published protocol to uh, extract the DNA contained for green algae or green seaweeds such as caulerpa, and how you preserve them, how you preserve the sample without reducing the DNA, the DNA concentration, because one of my students have a case and becoming a big problem uh, about her research is to extract the DNA from the fresh seaweed.
Yeah. Yeah. Yeah.
Yes, but from still now she cannot get the DNA with yeah, she cannot extract the DNA, so
Yeah.
she cannot get the results.
Yeah.
Yeah. Thank you.
I personally, I don't have a personal protocol, but it's not a secret. It's all the protocol is now available in the internet. So I just suggest to your student maybe to get the protocol from the internet and try if it works. If not, they can try another protocol. eh? So I think I think the internet would be a better source for protocol, because right now I'm not doing any DNA extraction in my lab. I'm not doing it. Okay. So if your student or you can have some further problem, please do not hesitate to write to me. Please do not hesitate to write to me. I will be happy to send you something also. Okay, thank you very much.
Okay, thank you, Prof. Lawrence, who already answered directly to Dr. Widya
Yeah.
Stuti.
Okay.
So, Dr. Widya, I think that's clear. So, I will now read some questions that addressed to you, Professor Lawrence, from the chat column. So, the first one is coming from Miss Rinta. She asked that, Professor Lawrence, in your experience and studies, what species of seaweed will become emerging species? What about, for example, laminaria, caulerpa, ulva, or gelidium? So,
Yeah.
Professor Lawrence, do you want to
Thank
answer
you. Yeah.
directly?
Thank you for that question. It depends what kind of emerging species that you are referring to. If you are saying, oh, emerging as environmental monitoring, then we have a group of species. Emerging species in terms of industrial applications, we have another one, or biotechnology. So the field is very wide open, really. The field is really wide open. And there are many. And of course, if you want to identify an emerging species, it has to be something that you can find. It's a species that is abundant in your country. So for example, in this question, you mentioned that laminaria, Delidium. Yeah. Laminaria is a cold water species. You cannot find it in Indonesia. So Caulerpa, I think, would be very good. It is emerging because there's a lot of things going on in that research of Caulerpa, antioxidants, and many other features. In order to answer that question totally, I will suggest that you just perform a literature search, and this will all come up. This will all come up. So the emerging species will depend on economic potential. It will depend on their utility. So these are the things. So it really depends what is your target. Okay. So I hope that is answered already. Thank you.
Another question, Professor Lawrence.
Yes.
This one from Mr. Jacob. So how can you revive 2,000 years DNA in seaweed preserved samples? It is because the DNA has got protected from the degradation by some substances in seaweed.
That can directly replace gasoline, diesel, even jet fuel and LPG, all of which can be produced by micro-refineries.
Somebody is answering the question. Correction, it's not 2,000 years, it's 200 years, okay? 200 years. Now, this extraction of DNA in old samples is actually a lucky thing. It's not guaranteed. It's not guaranteed because it really depends on the way how we preserve the sample. Luckily, in many seaweeds, you can get DNA even from 2 or 3 or 4 spores. You know, spores like pollens. So people are lucky in obtaining these samples from old samples and from really very small sample, old and small sample. So I think it is rather an exception. It is an exception, but it is not impossible. So you can still get these samples from herbarium specimens. 100 years, 200 years, I just keep on trying. But of course, obviously, the best samples are the ones that are freshly obtained.
Okay, thank you, Prof. Lawrence.
Thank
the
you.
next one, you have questions from Dr. Husnul Yakin, who is also lecturer in our faculty. So he has 2 questions for you. The first one, that you mentioned that stomata can be affected by its presence by carbon dioxide. Do you have data that stomata are also affected by other pollutants such as metals and pesticides? Are there any histological changes of the stomata that are affected by metals or pesticides or any other polluting materials? That's the first one.
Yeah. Thank you for that question. No, I don't have data myself, but I am sure there is data in the literature. I cited that particular example just to show that these things are possible. That stomata, plant structures, some histological structures can be affected by CO2. And these are also affecting some cells inside the seaweeds. But I'm not, I don't have data, but I respectfully refer you to the internet again for such a data. Okay, and I think it's a very interesting question to connect the stomata, the changes in the stomata with pesticides, with heavy metals. I think that's a very nice study. Thank you.
Okay, that's the first question, Professor Lawrence. The second one, still from Dr. Husnul, is the method of making herbarium for all seaweed is the same or different among the seaweeds?
Okay, thank you. Basically, it's the same. The most important step is to dehydrate your sample as soon as possible. Dry your sample as soon as possible, because if your sample become moist, they can be subject to fungal attack. So this is not a problem in Indonesia because the sun is so bright, it's so hot, so you can easily dehydrate your sample very easily. So there is a general process, dehydration. Some samples need to be air dry, and some samples need to be cannot be air dry, but they can inside the ethanol, for example, or you can put it inside a bottle with a formalin, for example. Okay? But then the standard method is really pressing and dehydration.
Okay, thank you, Prof. Lawrence.
Yeah.
The next one. So this one from Miss Irina Mildawani. So, Prof. Lawrence, if Padina species absorb poisonous materials, then it is very dangerous to consume it. And it means we have to be careful to consume alga that may be contaminated by pollutants. What do you think?
Yes, I agree. Well, fortunately, Padina is not consumed by people because it's not delicious. So there is no danger. Right now, there are many studies checking absorption of heavy metals, presence of heavy metals in Caulerpa, for example, because the Caulerpa is the latuk which we eat. And so far, I have not seen any data that suggests that there is a high level, a very high level of heavy metals and pollutants in the Caulerpa. So Caulerpa is not very good in incorporating the heavy metals into its tissue. So far, the data that I have seen is that the heavy metal found in the Caulerpa is below the WHO level. So it is not dangerous to the human health. And I hope it's that way. Okay?
Thank you
very much.
And then we still have one question.
Please, yes.
Yeah, because we also run out of time.
Okay.
But here we have sharing from Dr. Banda, who is also lecturer from Unhas.
Yes.
He mentioned that Padina and Sargassum are very common at our small island Riflat and Ulfa as well. We collect a lot of geotech photos of those algae. So Dr. Banda is actually quite active in collecting the geotech photos of these algae.
Yes.
And also, Lawrence, actually, we have also researcher from Unhas who frequently make herbarium from seaweeds.
Yes.
Unfortunately, today she is not attending this webinar because I know she's in the field right now collecting seaweeds.
Yeah.
Such unfortunate, but maybe I can connect her with you later.
Thank you.
Okay.
Thank you.
The last question, Prof. Lawrence. This is from Miss Elia Sinurat. So, dear Prof, your presentation was very nice and clear. So she wants to ask about climate change effect on seaweed. So she find a manuscript on the Asparagopsis taxiformis ability to reduce methane. How the mechanism Asparagopsis taxiformis reduce methane and also inhibits the global warming.
Oh my, okay. I don't know exactly how the Asparagopsis stops the thing, how it stops the global warming. But I know that some compounds inside the seaweed, when the compounds are inside the stomach of the cow or animals, it kills certain bacteria. It kills certain bacteria that produce this methane, that produce this high greenhouse gas. But as to the exact mechanism, I don't know. I'm sorry, I don't know. But right now, a lot of investigation is going on. And like how much should we give to the animal so that we can reduce the amount of methane by also how much. So there's a lot of things going on in that area. Also, there are many scientists now doing mass culture, mass propagation of Asparagopsis, because suddenly the price of Asparagopsis went up so much because they found out that, hey, we can put this in the feed for the cow and we can help save the environment. Okay? So now a lot of people are doing cultivation technology. They are doing the life cycle, which you can also do in your case, because Asparagopsis can be found in Makassar also. Right? So if we can make our basic research very strong, we can connect it with the applied research, like in mitigation of climate change and CO2 in the air. Okay? Thank you. I hope that answers your question.
Thank you, Professor Lawrence. I think the exact substances that uh, can inhibit the bacteria is bromoform, Professor Lawrence.
Okay. Yeah, that's right.
That one
Yeah.
can inhibit the bacteria and, of course, reduce the methane gas produced by the cattle. Okay,
Yeah.
I think that's the questions from the participants, Professor Lawrence.
Yeah.
It's very interesting talk and a very interesting discussion. Unfortunately, we don't have much time left,
Mhm.
so that's the end of our discussion.
Okay.
So thank you very much once again, Professor Lawrence, for answering those questions and for the great presentations. It
Uh-huh.
was a pleasure to have you with us here today, and you gave us also a take-home message
Yeah.
on how that the herbarium is actually not only for taxonomy, but
Mm.
also for other purposes.
Yes.
Another take-home message from you is that how we can care about our biodiversity.
Mm.
So we have the highest biodiversity,
Mm.
but unfortunately, we store it in a different country,
Yeah.
not in Makassar.
Yeah. Yeah.
So
Yeah.
I think this is the challenge, I think, and
Mm.
our students are attending the seminar, and I think our students, hopefully,
Yeah.
in 50 years, in 100 years, they can preserve our biodiversity by one small step by collecting the herbarium. So
Yes.
thank you very much once again, Professor Lawrence. Ladies and gentlemen, so this is concludes our presentation and discussion session. Thank you very much for attending, and we hope that you learn and of course you enjoy today's Top Scientist webinar, and we hope to see you in the next Top Scientist webinar. Thank you very much, and see you next time. I'm giving back the mic to the Master of Ceremony. Thank you.
Okay, thank you very much. Thank you to Professor Lawrence Manzanulio for the information and knowledge you gave on this joyful day. And also thank you very much for Jamaluddin Fitra Alam PhD for guiding this webinar very well. Ladies and gentlemen, before the closing session, we would like to share the electronic certificate that we have prepared for our distinguished speaker, Professor Lawrence. So please, maybe after we show the certificate, we will continue with photo session before we close this webinar. So please, Miss Isra, could you show the certificate, please?
All right. Can you see my screen?
Yeah.
Okay, thank you very much, Professor Lawrence, for your valuable knowledge and time. And this is our appreciation from the Vice Rector Hasanuddin University, Prof. Adi Maulana, because for the sharing your valuable knowledge to our student and lecturer in Universitas Hasanuddin. And don't be worried, we will send you this certificate by email as well from Dr. Jamal. We'll send it to you as well later.
Thank you very much. Thank you. Thank you for your certificate.
And then I think everything is awesome today. We finally come to the end of today's agenda. Before we officially close our webinar, we want all the participants to open your camera, please, because we want to take a photo session with our distinguished speaker, Prof. Lawrence. Okay, I will count in 10 seconds. Please, everyone, open your camera. All right, here we have X slides. So I will start in the first slide. All right. Yeah. All right. 1, 2, 3. Smile. Yes. Second slide. 1, 2, 3. The third one. 1, 2, 3. The fourth. 1, 2, 3. The fifth. 1, 2, 3. The sixth. 1, 2, 3. And the last one. 1, 2, 3. Okay. All right. I think we finally come to the end of today's agenda, and let us close the webinar very soon, alhamdulillah. And I will forward to Ms. Alia.
Okay, thank you very much for Ms. Isra. Ladies and gentlemen, we finally come to the end of today's agenda. On behalf of the committee, thank you for making time in your busy schedules to join us here. From the deepest of my heart, I apologize for my mistakes in presenting this event. I wish you all a pleasant day. See you on the next occasion. Wassalamualaikum warahmatullahi wabarakatuh. Thank you very much, everyone. Thank you, Professor Lawrence. Arigatou gozaimasu.
Yes.
Arigatou gozaimasu, Professor. Thank you very much.
Thank you, Professor Lawrence.
Thank you. Thank you very much. Thank you.
Thank you.
Thank you.
Everyone. Thank you,
Please,
Prof. Lawrence.
please keep in touch.
Yeah.
Okay.
Yeah.
Yeah,
See
you can share my email address to everybody.
Okay.
Thank
No
you.
problem.
Yes.
Thank you.
Thank you very much, Professor.
See you, Sensei.
Tapi Bapak Ibu untuk nanti sertifikatnya akan dikirimkan ke masing-masing email. Jadi dimohon Bapak Ibu semua mengisi link yang kami sediakan di room chat untuk agar kami dapat membuatkan sertifikatnya dan mengirim ke setiap email yang sudah tersedia.
Materinya bisa di-share juga, Ibu.
Kami nanti tanyakan dulu Bu kepada narasumbernya, mudahan kalau beliau bersedia nanti kami akan sertakan juga di dalam link yang kami kirimkan lewat email nanti.
Oke, makasih Bu, makasih panitia.
Iya, sama-sama Ibu.
Makasih panitia, makasih
panitia.
Sukses selalu.
Kita open videonya.
Iya Ibu.
Sampai ketemu.
Ibu Irma.
Linknya di absen itu kan, Pak ya?
Iya Bapak Ibu.
Link absen kan?
Iya Bapak Ibu, benar.
Terima kasih.
Iya.
Mohon lift.
Kami izin lift Bapak Ibu. Mohon terima kasih atas waktu dan kesediaannya. Mohon diisi link absennya dan nanti kami akan kirimkan email sertifikat dan materi insya Allah. Izin lift Bapak Ibu. Assalamualaikum warahmatullahi wabarakatuh.
Konteks MPS dan seterusnya, maka kalau itu dianggap setelah memenuhi, itu pasti akan menggetarkan sistem yang lain, akan beresonansi ke komponen sistem yang lain. Proses ini memang tidak langsung tiba-tiba jalan atau sinkron ya, tetapi paling tidak